To accelerate biological discovery, you need smarter tools. Tools that combine the precision of genome editing with the power of hybrid learning, digital twins, and sensors. This is the core of biointelligence. I am Marco Integhini, and this is Biointelligent, a podcast that decodes biotech scientific innovation within the Horizon Europe project BIOS and more.
All right, so today I'm joined by Tobias Sauer from Fraunhofer Gesellschaft. Fraunhofer, based in Germany, is one of the leading organizations for application-oriented research, with a strong focus on future key technologies and transferring research results into industrial practice. Within the Horizon Europe project BIOS, Fraunhofer is one of the partners helping us to turn biointelligent biomanufacturing into concrete tools and workflows. Thank you, Tobias, for joining.
Thanks a lot, Marco. Thanks for the introduction. It's a pleasure for me to have this be part of this interview podcast today. And yeah, I'm happy to talk about our technology, ELLI.
Okay, sounds cool. Tobias, I would love you to introduce yourself a little bit, specifically if you can tell us about your role in Fraunhofer and connect it to the BIOS project eventually.
Yeah, for sure. So my name is Tobias Sauer. I'm 30 years old now, and I'm an automation engineer by training. And I'm here with Fraunhofer for like 5 years now. So I'm quite experienced in being a development engineer for lab automation devices. So we do basically from small devices, technology development up to bigger production plants for pharmaceutical or medical issues. We do it all. And also in the BIOS consortium, I'm the project lead of Fraunhofer, especially of the work package 3.4, where we plan or already did develop automated electroporation platform that we call ELLI.
Okay, so you already developed ELLI, right? So you already have you're already quite advanced on it, and we will touch on that. Yeah, maybe for the broader audience who might not be so expert technically, could you describe what ELLI does and what practical problem is this platform designed to solve?
Yeah, for sure. Nowadays, we have in biotechnology a lot of strain engineering is done, but the problem is that like the parameter space is quite big to modify the strains genomically in the right manner that they produce biochemicals or other products that we want to use. So this production or this development of these strains is really time consuming, and especially when using electroporation, it's really time consuming because electroporation in a lot of cases is still really manual work to do. And also when we use there are some devices that also automate these kind of steps, but they use really specific consumables, which is also really expensive then. And for this strain development, in the end, we need fully automated screening platforms to rapidly screen different possible genomic modifications and to find the right modification in an appropriate time. Basically, this is what ELLI does. ELLI closes the bottleneck in high throughput electroporation, and it automates the process itself. So the electroporation process is parallelized and automized, and also ELLI can be integrated into these automated screening platforms so that nobody or no human is needed to interact or is really part of this production or screening process.
All right, thank you. Just to maybe elaborate a little bit on this. So the thing that makes ELLI different from other approaches is this like fully automated circle loop in a way. You mentioned no human intervention is needed. Can you clarify a little bit on that?
Yeah, so this means in the end that, as I said, the electroporation itself is automized, and we can implement it into an overall automated workflow. So, for example, the cell preparation will be done by a liquid handling robot, and then in a microjeter plate format, and then the microjeter plate will be handed over by a robot to ELLI, or even in a more simple way, the liquid handling robot is pipetting directly into the plate, which is on the nest of ELLI. So we have a smooth integrated automated workflow, and then the process or the plate is processed inside of ELLI, and afterwards the plate can be taken by a robot, and like downstream processes can be performed. So we have a fully automated workflow in the end, from the beginning, from the sample preparation through electroporation and until the plating or the screening of the transformed organisms in the end. Like the humans, they just move into an more overall work, because they don't have to perform the electroporation itself. Yeah, they're just supervising it.
All right. That sounds fascinating. It seems it could really simplify the day-to-day work of lab scientists who usually need to perform these steps manually. And well, could you confirm this? So is it also meant to help a day-to-day lab?
Yes, for sure. So the automation part enables a faster strain development in the end, because, as I said, the humans are not operatively active in the process; they're just supervising it. Also, with the technology of ELLI, we can perform 16 electroporations at the same time, which increases the time compared to the manual process with the cuvettes. And also the sterilization, which is included in ELLI, enables also the fully automated process. Thereby, we are able to just use standard labware, which which reduces the cost and also reduces the carbon footprint in the end, because we don't have to use 384 cuvettes for the electroporation, but just one plate.
All right. So well, that's more clear now. Why is that useful? So I wanted to ask you, what's the status of the device nowadays? So is it already used in other labs? Well, and also I wanted to ask you if if someone is already using it, and how was the process so far? Like, did you have to send it over to some lab to test it? Are you still doing it, and so on? So yeah, that was the question.
So during the project, we started with the technology development internally, and also with setting up a reference process here at the Fraunhofer to test the technology in fast iterations. And now we have a demonstrator, which the technology of ELLI and the core technology we developed is inside it. Yeah, so we have a device which is portable, and we can put it in the car and go to some labs and test ELLI externally, which is also our goal, because we just tested it with our reference process here. But in the end, we need to see if the real world applications are also feasible with ELLI. So we did this already one time. So we just last week we went to the University of Wageningen to test ELLI there, and it was a really successful test actually. So we
Nice.
proved that like the core functionality of ELLI is working. So the electroporation is working, and also the inline sterilization process is working. So we are in the end able to perform these electropораtions without cross contaminations between the different channels, and this, as I said, this leads to a less amount of the consumables that we use. And from now on, we also are looking for finding tech providers or other development partners to because ELLI at the moment is a demonstrator, right? It's not a product, but to bring it to the market, we need to find partners which are experienced in device development, and we want to have a partnership with them to develop ELLI filler and finally bring it to the market.
All right, but it's already a patent, right?
It's also patented, yes. It's It's a patented technology that we developed here in the project, and with a potential development partner, we are trying to get a licensing agreement then. Yeah.
All right. So, well, the study is really very promising, and yeah, that's great. Do you have any other project on this line that you are working on currently? Maybe not that advanced, but still, or something you would like to work on in the next future?
Yeah, I mean, we are working on improvements in this direction, yeah, like automated electroporation, also with other sterilization methods, because the one we are using has some points that can be adjusted or need further development, and we're also checking other technology possibilities. And also we are trying to think about what is before the process and after the process, down and upstream, which we can also automate. In the end, the vision is a fully automated screening platform, and I think the fully automated electroporation was not the only bottleneck in this section.
Okay, so you see some other potential issue to overcome in the next future, but the goal is to automate this process as much as possible. So the fully integrated platform. That's very cool. Yeah. Now speaking about automation and innovation, I wanted to ask you, this is something I also asked to Enrique in our previous interview. If you have a definition of biointelligence, what do you consider biointelligent?
So, of course, ELLI is part of this biointelligent ecosystem that we are trying to build up, because ELLI is not biointelligent itself, right? It's not direct combination of biology and technology, which is a sensor or whatever, but it's still inside this ecosystem, because we get to know more about the biological processes by enhancing technology to understand it and to make it visible for us as humans, and as I said, to automate the whole process even more and to reach faster development results compared to the current situation. In the end, we need the combination of these three issues. So biology, technology, and information technology, to get a more intelligent system and to understand better the biological processes, and also to use the biology even more for industrial purposes. Yeah.
All right. I guess I can be satisfied with your answers. Thank you very much for joining me for this interview, and looking forward to see you presenting ELLI in person in the next future. And all the best of luck for your project.
Thanks a lot. One thing I want to mention, you just talked about it, to get to know ELLI, which is we can, or you can, also the audience can visit or meet us in the upcoming fairs and events. So we will be in the SLAS in Vienna, and also in the Future Labs Live in Basel. So just come up and get in touch with us, and we are curious about other applications where ELLI might be also closing the bottleneck, like it did in this showcase here.
All right. Perfect. So I will also make sure that your contact will be visible in any channel we will publish this interview, so everybody can easily reach you if someone needs any information about ELLI and wants to learn more about it. Okay. Thanks you again, Tobias, and it was very nice to speak with you today. And yeah, looking forward to see you soon.
Thanks a lot. Bye-bye.
Bye.